So this is the second part of an overview on cancer and its biology. The last time we got together, we went through a series of talking points, ending at signaling pathways, and I'd like to finish off talking about tumor microenvironments, and then moving through tumor detection, cancer therapy, and resistance to therapy at the end. So to start off with, tumor cells live in a microenvironment that is always changing. It's always evolving, and it's evolving to the advantage of the cancer. And each cell type that's in the tumor microenvironment generates signals that drive cell mobilization and activation. And by those signals, I mean growth factors, chemokines, cytokines, hormones, and a series of neurotransmitters. Chemokines are a class of compounds that are secreted by white blood cells. Chemokines, on the other hand, are secreted by host cells, and they are intended to activate the lymphatic cells. They also have a bad habit of self-stimulating the host cells and making them grow faster. In addition to those growth factor related molecules, there's a series of other things that is in the extracellular environment. And that includes the extracellular matrix, things like collagen, which acts as a scaffolding and also is involved in signaling. In the local area, there's also soluble receptors and decoy molecules, metabolites, pH modulators. And I would like to note that tumors tend to have a relatively acidic environment. That's where they live. They don't have a nice, happy 7.2 pH that you associate with a normal cell environment, but rather they have a pH that is closer to 6 or even lower than that. And that's a big change. Normal cells actually have a hard time growing in that environment. Tumor cells do pretty well with it. So again, you have a slight advantage just due to the changes in pH. The tumor cells do live in a very complex microenvironment with many different types of cells just in the local area. Naturally, you have a preponderance of cancer cells, but you also have in that tumor mass immune cells of various subtypes. These are T cells, B cells that produce antibodies, natural killer cells. There's also macrophages and other cells of the immune system. In addition to the immune cells, there's also fibroblasts. These are fibroblasts are cells that end up making collagen, but they become associated with the cancer and start secreting a lot of growth factors. Endothelial cells are cells that make up the blood vessels. And of course, they would be in the local environment as well. Mesenchymal stem cells and adipose cells are also there. Adipocytes are cells that are fat cells, but they also supply a large amount of energy and food directly to the cancer cells. So they can have a very detrimental overall activity because they're feeding the tumor cell. The microenvironment evolves from the initial cancer event all the way through metastatic outgrowth. And it changes over time as cells undergo the process of breaking out of their local environment, migrating to a new site and invading the new site and becoming therefore metastatic. And that change in microenvironment can dramatically affect the type of therapy that is used to combat them. Oh, I got this one. We've seen this one already. There are some signals, pathways in the microenvironment that are relatively unique. These include the signal transduction pathways. These pathways are networks of proteins that receive extracellular signals like growth factors and translate them into cellular actions. That includes cell division, growth, survival. In cancer cells, one of the truly dangerous things about them is that their growth signals tend to be permanently switched on. They're in a state of hyperactivation with no way to turn the growth signals off. And the key molecules that are driving these are the oncogenes that become activated and result in excessive growth signaling. On the other hand, tumor suppressors, which we talked about last time, are inactivated. So again, the brakes are removed on the cell growth. So you have stimulation of growth and no way to turn off that signal. So I'd like to now sort of change a little bit of directions and end up going on in a slightly different direction and talk about cancer stages and ending up at metastases. I want to point out that as sort of as a transition from the last section, that tumor masses are biologically diverse. The center of a mass of tumor, which is typically called the core, and the periphery, which is the edge, have difference and significant differences in cellular composition. Their microenvironment is frequently very different. And they also have differences between the genetic and metabolic activity. So going from the edge to the center, things are dramatically changing in a tumor mass. The core of a tumor mass frequently contains inactive or necrotic cells, while the edge is typically more active and invasive. And in some cases, this can become quite exaggerated. So you have a tumor mass with just the edge containing live cells and the internal mass being totally dead. It can look something like maybe a tennis ball or maybe a golf ball would be is cut in half. Might be a better analogy. Therapies that target one part of this tumor may not be effective against the other because of the difference in biology and microenvironment as you proceed from the outside of the tumor cell, the tumor mass, to the inside of the tumor mass. This is a type of spatial heterogeneity, and it is a major factor in tumor growth and also resistance to treatment and ultimately metastases. Metastatic cancer cells are also diverse. So you have a mass and then you have cells within that mass. Cells within that mass are diverse. They represent a mixed population of cells. So this is problematic because you're not looking necessarily at one disease. You're looking at a multiplicity of diseases all within the same tumor mass. The cells vary genetically and phenotypically. That is phenotypically, I mean, in the way they look and act within a given metastatic site. And often the genetics of one tumor site varies from another site. So the tumors are not only metastatic, but their genetics is different, even in going from one part of the body to the other. The most predominant genes that are changing during metastases are ones that we have sort of mentioned before. Those include, but are not limited to, the tumor suppressors. And very importantly, DNA repair defects are common. So things continually to get worse because the DNA defects that spontaneously arise in a cancer are not being repaired.
Paul, could you take a second and go back to the first picture for microenvironment? One with the vasculars, yeah, that one. Yep. I just wanted to also share some concepts here. So Paul was just talking about how there is a morphology to the tumor, how there's the inner and the outer, how there is potentially a progression of, you know, how damaged things are, and also perhaps even the function and what's going on with the tumor at these different levels. You'll recall in part one, we talked about in some cases inside the center mass of a large tumor, you'll even get to the point of having necrotic tissue. In the case where it's not dead, where it is alive, one of the situations is what you see here, which is that vascularization. So there's some of the microenvironment is involving the recruitment of a number of things, including the growth factors for the capillaries. And this kind of concept of co-opting and recruitment is very common in the microenvironment. You know, there's this, you know, it's not just that you have a cell that is gobbling up nutrients. It is the survival of it is such that it is incentivized to release signals that co-opt the environment around it to help it survive, right? So to be more, either those could be growth signals, but they could also be metabolism signals, all kinds of co-opting signals. And when you look at tumor progression, for some of the tumors, there is a locus that the tumor can start to form. It's kind of like a precancerous locus that has some kind of disrupted microenvironment. So you can actually observe disruptions in the signaling chemistry around these cells, around where these tumors are going to form, and then that can kind of cascade. So think also that there is a progression where, you know, you could go from simple forms of recruitment that, for example, maybe there is like one cell that's the origin cell for a major mutation, but maybe some of those mutations are things that promote growth in the region. And then those things, because they are promoted to grow, are more likely or more susceptible to become mutated also, especially if you have those DNA repair genes, you know, that are already compromised. That TP53, for example, very common in most cancers. Its function is the DNA repair. And so if you have anything spurring cell division, there's kind of an increased propensity for that mutation. But there is that recruitment. And another way to think about this also, when Paul mentioned briefly that the tumors are a bit acidic, I would say that's a bit predictable. And one of the reasons is based on our past conversation. You'll recall that some of the tumors are doing is, yes, they are trying to recruit these capillaries, and they will grow to the ability for them to get blood, but also they will start to use anaerobic metabolism. That is metabolism that doesn't depend on oxygen, right? And so what does that produce? That's the same thing you do when you work out. You get lactic acid in your muscles. These tumors would presumably be using this anaerobic metabolism as well. So you could kind of predict, you know, or kind of, you know, see why tumors might be acidic. There might be other things going on. There's absolutely ion channels and hydrogen pumps and stuff. But I think metabolism of the tumors is probably a big indicator of why that might be. But the point that I want to make is that there is not only some kind of positive feedback in especially the more aggressive cancers that are releasing things that are beneficial for it, for its survival, but also triggering to nearby cells to perhaps mutate themselves. So there is some coercion that has been documented in literature. Not all cancers are doing it, but there is known coercion where you could have nearby cells kind of be shifted into something that's more or less useful for the tumors. And so this microenvironment, I would also say, is not as fully understood as you would hope. I think a lot of the narrative that we're going to see is around the cell signaling and understanding what's going on inside the cell that's mutated. But I would say that a lot of active research is very much looking at the role of the microenvironment. So I would call that out as ongoingly important, understudied in literature, but very important every time it's looked at. So with that, I'll yield back to Paul. Thank you. Actually, before I do, are there any questions so far? Okay. All right. Go ahead, Paul.
Okay, so we've been talking about metastases, so I thought it would be useful just to talk about it in a little bit more detail. Metastasis can be defined as the spread of cancer cells to other parts of the body. It involves a series of very definite steps and stages. The first involves detachment from the primary site and a local invasion. Once that occurs, there is intravasation. That is to say, the cancer cells enter the vasculature or bloodstream. Sometimes they also invade the local lymphatic system and can end up moving via that process. That movement is called dissemination, where the cells travel through the body and they potentially settle in distant organs, although they can also metastasize to a different area of the same organ. Typically, they can adhere to the walls of blood vessels or lymphatic nodes. And once they attach inside a blood vessel, they extravasate. That is to say, they get out of the bloodstream or the lymphatic tissue, and they enter the target tissue. And that leads, of course, to colonization, where the cancer cells establish a new tumor in the target area. So this is just sort of a picture of that, where cancer cells enter enter from the primary site, they get into the bloodstream, and they move through the bloodstream, typically until they get stuck in one of the capillaries. And once they get stuck in a capillary, they can end up then extravasating and getting into a new site and causing the metastases. I should point out, since we've been talking about genes throughout this, that the gene alterations that drive metastases are actually different in many ways from those that drive tumor development, although there is frequent overlap. There is a number of frequently mutated genes, which we've talked about before, that you find in metastases, and those can include TP53, but also some new players, the ones that we haven't really talked about very much, including EGFR, which is a growth factor receptor, KRAS, which is involved in signaling, and a few others. Metastases drivers include a different set of genes. I'm not going to go just read through the list, but just to say that they're different. And they end up producing different molecules that have different biological effects. And importantly, metastatic seeding, movement from one site to the secondary site, happens very early and well before it can be detected by the patient or can be detected clinically. So typically what happens is when a person gets a cancer, they will feel a lump and they will go into the doctor and say, I have a lump, can you take a look at this? Or they'll have some other factor that leads them to go and visit their physician. But the primary driving event that caused that metastases typically occurs almost 3 years before. During that 3-year period, the tumor is at the metastatic site is quote unquote silent. It's not detectable. There are several stages that are clinically defined for the spread of cancer. Stage 1 is where the cancer is confined to the organ of origin. Excuse me. Stage 2 is where it's locally invasive. Stage 3 is where you have spread to the lymph nodes and sometimes bloodstream. And then you have stage 4 where you have spread to distant sites with pronounced tumor development. And regardless of what type of cancer you're talking about, those stages will be always very similar or identical. This is something that I put up that is reflective of staging of cancer as the 4 stages. And the primary reason I put this up is to give you an idea of the size of that tumor mass. So at stage 1, it's typically around the size of a walnut. Stage 2, where you have migration to local lymph nodes, the size of the tumor is about a large walnut or a peach, up to that range. Stage 3, the size is about of a lime. And then stage 4, the tumors are actually pretty big and can approach the size of a peach. So with that, I'm hoping that you get some idea of how large the tumor masses can actually get as you go from initial clinical diagnosis to a stage where things have become quite life-threatening in stage 4. I couldn't be complete without showing you a couple of histology pictures. So this is what they call a section. So a small piece of tissue that comes from the adjacent lung. So this is relatively normal lung tissue. You can see that there's quite a lot of areas where there's air, as you would expect in the lung. And adenocarcinoma, on the other hand, so you're looking at 2 colors here, one of which is pink and the other is blue. The blue doesn't show up too well. I'll show you something that shows it a little bit better in a minute. The blue represents the staining of the nuclei. The pink is associated with the staining of the cytoplasm and extracellular matrix. This pattern here is very dense compared to this. There isn't a lot of air pockets in here. There is some mucus accumulations. But this pattern is a solid nest of neoplastic cells, which is accompanied by some of the mucus areas that I mentioned earlier. Little higher power lets you see the blue nuclei more distinctly. Again, what you see in this small cell lung carcinoma is very dense tissue. The nuclei are quite round, but the cells themselves are actually very small, are relatively small in terms of their overall size, and impart the name of the carcinoma itself. Non-small cell lung carcinoma has a different morphology, the way it looks under the microscope. So you see very pronounced and irregular nuclei, and it doesn't show up too well here, but the cells are actually quite elongated. You see some of that here.
So I want to be very clear. These are both called lung cancer. Now, what Paul is saying is that the shape of them is different. And it's not just that the shape of them is different. These are actually different cells within the tissue. You know, the tissues are a collection of cells. Some are structural, some are cleaning cells, you know, mucus producing cells. So these come from two different types of cells in the lung. Is that right, Paul?
Yes.
Mm-hmm.
And that's sort of illustrated here. In that there are two primary, if you're a smoker, there's one primary area that you need to be concerned with, and that is the primary bronchi and the secondary bronchi, where this is where smoke impacts physically directly onto the walls of your breathing tubes. And it does that typically in these first few bifurcations. And that is typically where you end up getting non-small cell carcinoma and from smoking. Non-small cell lung carcinomas actually come from a slightly different region that is deeper down into the lung and is associated with some glandular-like structures in the normal lung. And then there is another transition zone down to the very deep lung where the carbon dioxide and oxygen exchange actually takes place. And you have a switch again of these tumor types. So where you are in the overall structure of the lung makes a big difference in what type of cancer you end up having. This is similarly true for other types of cancers. Breast cancer gives you, there's branching that occurs in the breast, and typically you can get different regions associated with different cancer types. So this is sort of a common theme in cancers. They depend on where they are in a given organ and what their role is in the overall normal biology.
Paul, if I could interrupt here, could you please go to the stage 1 through 4 picture? So the emphasis with what we, this part, and especially like the stage 4, these are the metastatic cancers. Those are the ones that the emphasis is on the progression out of one tissue into a different tissue, or this that invasion that we were talking about. It is important to be aware that when we talk about tumors, there's of course entire disease sets that are just focused on benign tumors. So a lot of what we're focusing on right now is the metastatic cancers. But be aware that when we talk about tumors broadly, there's also benign, like neurofibromatosis was benign tumors all over the body, but again, benign. So the emphasis being that there was no movement out of those tissues. Wherever your tumor was developing is kind of stayed there. Happens to be that it was formed independently all over the body in the NF1 type disorder. But there was none of that, like now it's going to penetrate into the bone kind of emphasis. And Paul, if you could please go up to the slide with the vein, just 2 or 3 slides up. I want to make it clear, we were talking about, what has to happen for a cell to become metastatic, right? And so if you think about what Paul was talking about, there is a leaving of a cell or a cell cluster from a collection of cells, and some kind of, normally cells are bound together in the intercellular matrix. That's the collagen and the microtubules and all these, the gaps, and they're relatively tight. You don't really have jiggly cells from most tissues. but we're talking about something that's able to leave, enter the bloodstream, and at some point actually enter out of the bloodstream back into the body, or being stuck in that capillary. The type of cell that is doing that, that's going into the body, that's actually a relatively mobile cell. Not all cells are evolved for that level of mobility. Not all types of cancers are candidates for becoming malignant, or at least not as often becoming malignant. Neurofibromatosis type 1 is one of those that it can become malignant, but it's not as common. It's maybe 20% or something. But that's 80% of the tumors that never become malignant. And if you think about that, part of what we're talking about here is the ability for the cell to change how it interacts with its microenvironment. Where is it getting its metabolism, and what is it recruiting for motility, mobility? The way I think about it, I don't know how accurate this is, but if you think about cell differentiation, this is going from a stem cell that can evolve into different types of cells that have specialization. So one example of a very specialized cell is a neuron. There's a neuron in your spinal cord. It's a single cell that goes like 4 feet. Okay, so you have a very, very, very long axon. And that type of cell is not a good candidate for mass cell division, right? Like you're never going to have this 4-foot long cell dividing. In fact, it's a type of cell that once it doesn't even regenerate very well. You grow it when you're a kid and you kind of keep it. There are other types of cells that are more able to do cell differentiation. Now, when you have these cancers and they are accumulating these mutations, they can revert from a very specialized cell. Maybe it's a kidney cell, really good at extracting toxins from the bloodstream and releasing them into your bladder. It can change from that type of cell to maybe kind of almost a more ancestral cell, which is that more stem cell like. That's not exactly what's going on, but the concept is valid. Of you're losing that morphology. What if you're a very structural cell? You're kind of reverting to one that has the attributes of this more stem cell like nature, where it has motility, it can move around the body, it doesn't need to be tied to an intercellular matrix. All that is to say, there is a biological reason why some tumors are more likely to be malignant or less likely to be malignant, somewhat relating to what kind of cell did it evolve to be, what is its dependency, does it need to be bound to something from a cytoskeletal matrix? Some things never become malignant, probably based on what type of cell and tissue they were kind of differentiated into when they were becoming a tissue. But there's also counterpoint reasons why certain types of cancers are so deadly, are so malignant. I believe glial cells and brain cancer are one of those very easy to become malignant type of cancers. Is that right, Paul?
Actually, I don't know.
Brain cancer, I think it's glial cancer is one of the most, well, there's a number of reasons why it's so deadly. It's hard to treat, it's in the brain, but I do believe one of the reasons is that it's very easy for it to become metastatic. The glial cells are not the neurons in the brain, they are the cleaning cells that are around the neurons. They provide health, nourishment, cleaning, clearing of the brain. So they're not those highly morphologically structured cells like neurons, which are hard to divide. Rather, they're these smaller, more spherical cells that are easier to become those invasive metastatic cancers. So, there's a logic to why some cancers are more metastatic, and it has to do also with the cell signaling, of course, but it also keep in mind what type of tissue was it, where did it come from, what was its original microenvironment, and what microenvironment did it become once it was more and more malignant or cancerous. Before we yield, any questions for Paul or myself? Aria, is everything clear so far?
Yeah, thank you.
Okay. All right. Well, happy to just interrupt us if anything comes up. That's not a problem at all. Paul, go ahead if you want to resume.
Okay, again, using lung cancer as an example, I just want to point out that typically what happens in a clinical setting is that small cell lung carcinoma is not usually detected until it's already at stage 3. It's already metastasized. In small cell lung carcinoma, you have something similar, if not worse. Typically, clinical detection is at stage 3 or stage 4. And there's a very unfortunate finding having to do with survival rates down here and staging. So if a lung cancer can be detected when it's localized, you have a much higher 5-year survival rate than when it's distant. You have metastases, the survival rates are pretty dismal. I mean, there's no other way to say it. They're pretty dismal. So metastases is a critical event, and also its detection in the clinic are critical events in terms of how to address it clinically and how to try to cure it. Well, where do these tumors go? Where are they ending up in the body, all of these metastases? Well, we talked about one of the places is the local lymph nodes, or it can also be a distant lymph node. The brain is a very common site, as are bones, liver, adrenal glands, and there's a lot of other parts of the body. So this is the percent survive. These are survival curves depending on metastases and where they where they arise and what tissues they arrive. You note that all the curves are not at all favorable in terms of the patient's survival. They're pretty much all the same regardless of where the tumor ends up metastasizing. Lung cancer, by the time of the metastasizes, again, the 5-year survival rates are really dismal, in spite of all the advances that have been made in cancer treatment.
If you could go back just briefly, I do want to define the lymphatic system just as a reminder for people. This is a set of tubes in the body, and there's lymph nodes in between those tubes. It's generally involved in fluid balance. So we're talking about certain tissues have certain concentrations of water or lack of water, but also it's involved in the immune system. So this is one of the ways that white blood cells move to get to infection sites around the body. So it's kind of parallel, or in addition to the purpose of the circulatory system. And you can kind of see very logically why certain sites are commonly the secondary site of the where these things are being transplanted to, because they are moving. When you talk about the colonizing tumor cell clusters, they're moving through some kind of conduit or through some kind of system. So the fluid regulation system and the lymphatic can be hijacked for that, and then likewise where the blood flows are going. And then when you look at these tissues, these are areas that are specifically involved in aspects of those fluid or blood regulation. The brain is massively, it's the #1 energy consumer in your body. It's massively consumed there, but there is the blood-brain barrier, but still it's a big thing. Bones are involved in blood cell production. So there is a lot of actually fluid and bone interaction. We're talking about the marrow inside the bones in particular. Livers are detoxifiers. They're removing things. They're very intimate with the blood system. The hormone systems, most hormones are spread through the circulatory system. So all of these places where you're getting these metastases are very much related to and are logically predictable from, what kind of fluid interactions that colony was having. Go ahead, Paul.
So perhaps one of the most logical questions is, well, why don't we just screen everybody for lung cancer? Do it very early. The simple reason is that there's no good test out there that's available for it. The only recommended screening test for lung cancer is a low-dose computed tomography. So here you're using low-dose X-rays to look for a cancer. And typically when this is done, it's done usually from head to toe. So the entire body is getting in spite of the fact that it's called low dose, it is a pretty significant dose of X-rays. X-rays, of course, can be linked to developing your cancers. So it's a double-edged sword. This type of lung cancer screening is really only recommended for adults who are at high risk of developing disease because of their smoking history and age. So while it is used, there's big caveats associated with it. It has a very significant radiation dose, and I won't go into the numbers or what they mean, but it's significant. And I thought it might be worthwhile just to show you a picture. This is an image from a CT, a computerized tomography. And they're looking through a cross-section from head to toe. So these are just slices going from head to toe. This is about the middle of the lung. These dark areas here are actually lung tissue. And what you see, this area here is a cancer. You can just look at it, you can see that there's a big difference between the density of the lung, which has got a bunch of holes in it and makes it translucent to the X-rays. And this, remember the picture that I showed you where everything was very close together and compacted in the lung cancer sections, the histological sections. That's the same thing that happens here. They're dense. And the X-rays get blocked, and consequently they show up in a different density in the image. This is what a radiologist might see, and this would be a pretty well-developed cancer. So this is actually taking advantage. The imaging is taking advantage of a core property, biological property of the cancer to do the actual imaging. So just a bit on computerized tomography. If you're not familiar with it, it uses a combination of X-rays and computer technology to produce images from inside the body. You have an X-ray beam that moves around in a circle around the body, so that you end up getting different views of the same organ as the detector spirals around the body. The X-ray information is sent to a computer and undergoes computer processing to produce two-dimensional cross sections or slices. And that slice goes, you could start at the top of the neck here and just go in slices down, or do it this way, going from one side to another. And that way you can get good three-dimensional information on where the tumor is located and what it actually looks like. So this is typically done not only for detection, but also for surgical planning. But this is very heavily driven by computer interface. This is another type of diagnostic imaging that is used for lung cancer and staging. The images here are from something that's called positron emission tomography. As an aside, can anybody tell me what a positron is? Anybody from the student group want to venture what a positron is? No is
Um,
okay.
is it like the kind of like the opposite of an electron? Like electrons are
Yes,
the focus.
absolutely. Good for you. It is a form of antimatter. It is an anti-electron. And the really, really neat thing about having a positron meet an electron is once they come together, they will annihilate each other and release a large amount of X-rays or energy. And curiously, one of the really neat physics things about them is that they will release that energy going in completely opposite directions, 180 degrees different. And it's a high energy emission. So you think about it, now you have an X-ray going in two different directions. That really helps in terms of the imaging, because if you know where one electron or one gamma ray is being emitted, in what direction, you know where it is going out on the other side. And that way you can help locate in three-dimensional space where that tumor actually is. Typically, for positron emission tomography, they use a radioactive tracer. This tracer is called fluorodeoxyglucose, glucose being sort of the operative word here. And the radioactive tag is fluoride, fluoride 18. So in the last talk, I mentioned that tumors have actively used glucose as their energy source, and Matthew sort of referred to that earlier today as well. This is how clinicians end up using that biology. Fluorodeoxyglucose is taken up as a glucose analog into the tumor, and it stays resident there in the tumor. And it does so at the preference of the rest of the body, which is not taking it up anywhere near to the same extent. That differential between the tumor tissue and the normal tissue is what allows physicians to be able to detect and see the difference between the tumor mass and the non-tumor normal tissue. Anyway, just a little sidebar.
If we could pause at that last slide for a second. I want to plant a seed in your brain as young researchers. This is something that I've come to appreciate recently, and it's a keyword called biomarkers. And biomarkers are any of a set of, testable diagnostic criteria that are telling you something about either the disease or its progression. And these can be, for example, blood tests. It could be saliva tests. It could be reading your genome. It could be extracting cerebral spinal fluid. And a lot of the modern cancer therapies are trying to use biomarkers more and more. So what Paul is showing with the imaging, that's the standard, right? That is, in fact, everyone's going to get that regardless, right? If you have severe cancer, you're getting imaged. That is the gold standard. But there is a less invasive role for trying to use other biomarkers. So, for example, we have a collaborating partner that's looking at prostate cancer, and they're looking at blood tests that can predict if you have prostate cancer. And it's the same thing Paul was saying, it's very hard to detect at early stage. If there was a blood test you could get, great. And in fact, a lot of, we start to talk a little bit about another buzzword, which is personalized medicine. You're kind of asking what's unique about your tumor, what's unique about you. You have certain cells that were mutated, certain signaling pathways that were mutated, and then you're going to have certain signals, right? And one of the roles of biomarkers is if you have certain signals, that might mean that you're going to be more responsive to certain therapies. You have biomarkers that might say that certain types of pathways are being used, and if you don't have those signals, then maybe those pathways aren't being used, and therefore like those therapies might not be effective. And especially when you don't want to mess around, you don't want to, you know, you only have so many chances at chemotherapy before many months have passed. You know, the more accurate you can get with what you think you're prescribing someone, that's clinically very relevant, but also very related to drug development. A lot of drugs and the manufacturers are very incentivized to find what biomarkers are good indicators for how successful their drugs are going to be. So maybe you have a non-small cell lung cancer uh, uh, compound, but when you go to pass it through the FDA, you specify you need this one genetic mutation. And so if you have non-small cell lung cancer without that mutation, that's not what the FDA approved that compound for. And that's in fact not necessarily what the company wants you to use it for. They approved it for a very specific type of biomarker. And that's probably because it's known to really work well in the case where that exists. So I just want to kind of expand your horizons a little bit. Biomarker is one of those key words, and it relates to having a more intimate understanding in quite a broad set of systems, but potentially less damaging types of things. Again, these imaging, lots of radiation, etc. But things like taking this imaging and factoring in a genetic analysis and factoring in, you know, a blood sample or something like that. Those kinds of trends in cancer research are very modern and contemporary. So keep that in mind as well. But again, we're showing the imaging because that is still the gold standard from a clinical setting. Thank you, Paul.
Yeah, so if I'm a cancer patient and a doc comes in and says, I want to give you this dose of radiation to find the tumor, then I get therapy, and the doc comes back and says, okay, well, I want to give you another dose of radiation so I can find out how the tumor is doing. And he starts doing that every couple of weeks. You know, I'm going to get pretty resistant as a patient to that repeated invasive diagnostic. On the other hand, if the doc comes in and says, I want to monitor your tumor growth just by taking a blood sample, it's much easier on me, the patient, and it's much easier and less cost intensive for the doctor and the hospital. So there's a tremendous number of advantages to biomarkers. And Matthew is correct. It's a cornerstone and will continue to be a cornerstone of disease monitoring.
Also, when it gets into the realm of computational biology, a lot of the computational work is around finding biomarkers, either from large transcriptome or large proteomics data sets. So a lot of it is finding biomarkers and kind of putting a big story together. So there's this kind of big data angle to it as well. But again, could be very specific. Maybe there's just one thing in the blood you're looking for.
Okay.
Very cool.
So assuming that we can detect the cancer and continue to use lung cancer as an example, you know, there are several options for a physician in terms of how he's going to address trying to get rid of that cancer. Surgery is typically the first line of defense from a physician perspective. If you can cut it out, do it. That's not always possible. Sometimes the tumor has already spread and has become metastatic or has moved to an organ that could, if it's removed or damaged, could be life-threatening. So chemotherapy using standard approaches is frequently the second layer of therapy. That may or may not include radiation, where high-energy X-rays are used to damage or kill normal cells. Both the chemotherapy and radiation depend on the body's ability to repair itself more effectively than the tumor. More recently, and by more recently, I'm talking about, maybe the last 40 years, but more so recently, precision medicine and targeted therapy has become a very hot area where medical treatment is targeting cancer-specific genetic mutations, which Matthew was mentioning. And it's done not necessarily to kill the tumor, but to at least slow down the tumor growth. And if you're lucky, be able then to kill the tumor cells via some other combination or via some other combination with another drug. Immunotherapy has been a long dream in medical treatment. There are a few, but well-established immunotherapy protocols. And here what you're trying to do is get the patient's immune system to fight against the cancer and ultimately destroy it. Immunotherapy is not as widespread as most physicians would like, and is still sort of in the early stages of functional development. Treatment options typically change with tumor progression. So as I mentioned, physicians will usually start off with therapy and then go to more and more advanced treatments, depending on whether the patient actually has a limited disease or a more extensive disease.
On that note, think about it this way. Benign cancer, malignant cancer, benign may not be life-threatening, right? It may be, but it may not be. And the chemotherapies are toxins, right? We're talking about killing cells. It's going to kill normal cells too. Do you merit that level of toxicity? It's going to depend on that disease state.
Matthew's right.
I had something else too. You go ahead, Paul. It'll come to me.
Okay, all of these chemotherapy drugs here, every single one of them is a poison. They're nasty chemicals, and they depend on the ability to preferentially kill off the tumor cells. But they're nasty. They affect every system in the body. So eating is disrupted, sleeping is disrupted, hair falls out, can't go to the bathroom, can't pee, lots of side effects, and they're all very typical. And it is one of the, of course, one of the most unfortunate aspects of chemotherapy is the side effects and things that drug manufacturers are working very hard to directly address. The quality of life during treatment it can be a little bit rugged. It can be more than a little rugged. Just a classification of common chemotherapeutic agents. So there's alkylating agents. These are chemicals that directly bind to the DNA, and via that process, they inhibit normal DNA repair. There's mitotic spindle inhibitors, which affect how the cells divide. There's anti-metabolites, things that are analogs of what the cell uses to make DNA. And once those analogs get into the DNA, they disrupt DNA repair mechanisms. Topoisomerases are another type of repair molecules that are typically used. Doxorubicin, which you may or may not have heard of, is probably the most widely used chemotherapeutic agent in the world, and it is a topoisomerase inhibitor. And it's also very widely used in lung cancers, as it turns out. Okay, these are just some names.
Yeah, just to interrupt briefly. So what I wanted to give you guys was a qualitative sense on outcomes and chemotherapies. And so when you talk about a drug is approved for cancer, you would think it would be related to survival or remission, right? Now, sometimes success just looks like it stops growing. So, ideally, we're talking about a tumor that shrinks, right? Shrinking chemotherapies are actually not the most common type. A lot of chemotherapies, we're calling it a win if it stops growing and you have maybe a 3 or 5 year extended life expectancy. So if you look at, there's definitely like you're cured of cancer, there's complete remission, that's great, but some of the more aggressive cancers, the drugs that are approved from them are increasing your 5 year survival from like 5% to 30%. And that 30% survival increase is a successfully approved drug. Like that's what we have to offer from a scientific basis. So it depends on the cancer and it depends on the progression, but there are sometimes very negative outcomes even when you're having a successful response. And again, either because you become resistant or even if it stops growing, you could have other things going on that could still end up killing you. And so there's a lot of kind of bad news. I don't want to give you the impression that all chemotherapies shrink and eliminate cancers. That's nice when that happens. But you kind of have to look at what the drug is actually approved for, and especially going back to their efficacy studies to see, how much tumor volume decrease was there? Was it like a 5-10% decrease? It might not be complete remission for all types of therapies. Go ahead, Paul.
Okay, so with that in mind, there's a lot of work to do left to be done in the medical field. It is not lacking for things to do and problems to solve. They're out there, and there's a lot of patients that could directly benefit from improvements. And hopefully we're looking to you guys in the next 20 years or so to help address that. Anyway. Okay, we sort of talked about this before, in that targeted cancer therapy is looking to detect molecular abnormalities or weaknesses in cancer cells and minimizing damage to normal cells and healthy tissues. This is one of the key advantages and one of the reasons that targeted cancer therapy is becoming increasingly popular and very much of interest, is because it minimizes damage to healthy tissues. That is one of the key components of it. Minimizes the damage, doesn't eliminate, but minimizes the damage. And what it's doing is interfering in specific pathways or targeting specific proteins that are essential for cell growth. And it's intending to block the activation of cancer cells by growth factors, inhibit the formation of new blood vessels, interfere with signaling pathways that cancer cells typically use and normal cells don't use or use as much. It can, of course, damage or destroy cancer cells directly, but that's frequently not the way that most of these targeted therapeutics work. Some of them do stimulate the immune system to be able to address cancers, but the bulk of them are actually working up here. These three things: blocking the activation of cancer cells. Okay. We talked a little bit about biomarkers, or Matthew did. You're looking for the result of changes in tumor DNA. And a biomarker can be anything that you can detect. So it can be a protein that is secreted into the bloodstream, for example. That can be a biomarker. If it's secreted into the bloodstream, you can sample the blood and use very sophisticated techniques to detect the protein of interest. There are products on the market that are looking into proteins that are and cellular elements that are secreted into the feces or into the urine, and using those as biomarkers to help detect and monitor cancers. All of these are fair game for different types of cancer. So looking into the feces, you're looking specifically for changes that might occur in a colon cancer. In looking at urine, typically that's done for bladder cancers, for example. And you're just taking sputum is another one. Spit is frequently used to look at certain lung cancer targets. Those are ones that just come to mind right offhand. But it's becoming, again, very important, especially in terms of doing targeted therapy.
Some of the interesting things, there's a lot of really cool innovations, not just on cancer treatments, but on biomarkers. As again, the case is very simple. If you can detect it sooner, you can treat it sooner, your outcomes are better. And just likewise, I mean, if you can just monitor the progression, your outcomes are also better. There's a lot of biomarker assays and biomedical devices and testing systems that are very innovative that you can also get into from a research career perspective. One of the ones that's kind of new that I learned about a year or two ago, there are DNA fragments in the bloodstream. So if you imagine the tumor is massively dividing, it is also growing and dying and killing a lot of cells, and all of those, like when a cell dies, it explodes and it releases a lot of that intercellular contents into the surrounding areas that ends up in the bloodstream. So whereas DNA is too long to survive in the bloodstream, they are able to detect DNA fragments. And so Paul mentioned in the bloodstream proteins, but I'm also calling out a very interesting one was DNA fragments as well. And so there's all kinds of these really creative and innovative biomarkers that are just now hitting the market and really changing the game on diagnostics. So if you have more of like I want to be an engineer or even a chemist, you know, there's things on that side of cancer research as well that's very interesting and upcoming. Go ahead, Paul.
Okay. So from a very practical, patient-driven point of view, there are some real advantages to targeted therapy as compared to chemotherapy. And this has to do with survival. Patients that get put on targeted therapy, perhaps even in combination with chemotherapy, live longer. So these are two survival curves. From targeted therapy, patients are living longer. This is months. Many of them are not dying. Standard chemotherapy, you have, again, a very rapid decline in survival. And it is well below that you see of targeted therapy. Targeted therapy is not curing 100% of everybody, but it certainly is better than chemotherapy. And from a patient point of view, you look at this difference and you say, yeah, I want some of this stuff. It's not hard to convince patients that this is the direction to go in, and nor is it hard to convince physicians if the evidence is actually there. And that's developed during clinical trials.
I do want to pause there. For me, it seemed like, well, if immunotherapies are one of the most recent generation of cancer treatments, why don't we all just go into immunotherapy research? I think for me, it was a very philosophical thing. Our adult group has almost exclusively focused on small drug molecules, small molecule compounds. There's other classes, you could have biologics. So these would be like protein products, things like botulism toxins, things like that come from like Botox is one of those. And then you have the immunotherapies and other kinds of therapies. And I think what I've come to understand is that there's a place for all of them. Now, what I've heard, and I haven't heard much from immunotherapies other than, yeah, they're really great and they can be very effective for some people. But the key is for some people. I have heard, for example, that 30% of people respond to immunotherapies. Well, that's great. If you respond, then your body is, fighting itself and it's very healthy and that you can have true, complete remission. But what if you're that 70% that's non-responsive, right? And so there is still a role for the classic chemotherapies. We kind of have maybe some go-tos that you're going to want to try first. And then if those don't work, maybe you try some of the more experimental or second line, medications. But I do want to give you a sense that sometimes some therapies are known to be relatively effective, especially if you have certain biomarkers. Maybe you have this 80% response rate if you have, a specific set of really well-known biomarkers. But, not all cancers are as well understood as that. You don't always, like doctors aren't always testing for all biomarkers. Some of that's very cutting-edge research, or it's not afforded for some reason. So there's multiple reasons why you might need to have classical chemical-based therapies, even in the era of immunotherapies, right? Where you're not always going to be responsive, and that's going to be a recurring theme in cancers. And if you think about it again, in part because these cancers are adaptive, right? Or their mutation profiles is technically it's going to be unique per person, and even within the cell. Within the tumor, you have morphological zones, and you're going to have genetic mutating zones as well, right? You're going to have different colonies within the tumor that have different response profiles, and that just means that, it's very complicated. It has to do again with how far along in the disease. If you treat the tumor earlier, not only is it less likely to be malignant, but you may have accumulated less mutations. And therefore, you may be more responsive to a compound. It's possible that if you wait 4 months, then all of a sudden you've accumulated a colony of cells that have additional mutations that mean you're no longer responsive to a therapy that you had been previously responsive to. So there is a moving target with these therapies as well, where something can work earlier in the disease but not later, it just depends. There's a lot going on there. So active, robust research in a lot of types of therapies is important. I would say that it's a little bit, from my perspective, even chaotic. If I was a clinical researcher, maybe there's too much going on. But ultimately, hopefully, if you become specialized in a field and you really know this one cancer really well, you have a sense on what are the common line treatments, what are some of the emerging treatments, and you're keeping an eye on how relatively effective are they. And at the end of the day, a lot of what these doctors are just going to try and make a best effort. If it works in 80% of people, they're going to try it on you, right? You may be that 20%, but they're not going to know that yet. Or maybe there's a way you can develop a test to differentiate who will or will not be responsive, in which case, great, develop the biomarker test so that you can differentiate. But that's where a lot of the research is. It's not just one and done for these therapies. There's also maybe a progression and some investigation on what might work. Go ahead, Anushka.
Sorry, I don't mean to interrupt, but I do need to leave a little bit early for this meeting. So I just wanted to let you know. Yeah.
Okay. Thank you so much for joining. I think we have like 5 or 6 slides left, but you'll have the recording made available.
Okay, great. Thank you.
Thank you. All right. So Paul, I'll let you continue.
One of the things that is not usually talked about, especially when it comes to immunotherapies, is the cost. one of the downsides of immunotherapies is they typically are not covered by insurance. And hence they're underlying that is the fact that they're tremendously expensive and much more expensive than targeted therapies. And it's one of the things that next generation agents want to be able to address, is to drive down the cost of immunotherapy, because they're just 5 to 10 times more expensive than conventional therapy. And because they're not covered by insurance, the financial burden falls entirely on the patient, which is no fun at all. A lot of these cancers, as we pointed out, become resistant. You know, they're not, you use a chemotherapy agent or some other type of agent, they don't die. This is responsible for 80 to 90% of the cancer mortality. So you're getting resistance in the chemotherapy, and the cells just continue to divide in spite of the fact that they're being treated with an agent that should kill them. The resistance can be intrinsic, it can be traced all the way back before treatment, or it can be acquired during or after treatment. So the cancer therapy agent can act as a selecting agent, weeding out all the guys that will normally die, giving rise to resistant populations. When that happens, the tumor cells become less susceptible to not only that drug, but typically other drugs as well. And that can occur from a number of different reasons. These cells can spit out, that is to say, they have enhanced efflux of the drugs. Tumor cells have developed, and normal cells too, have developed mechanisms when they meet a noxious chemical. Once they get it inside the cell, they will just pump it out. Cancer cells have that same mechanism, but on steroids. They will see a cytotoxic agent, a chemotherapeutic agent, and they just pump it outside of the cell, so it's not hurting the cell. They have elevated levels of metabolism that destroy the toxin once it gets in. They have increased, in some cases, increased specific repair capacities. They secrete growth factors to help stimulate themselves into continuing to grow. They have changes in their own genetic makeup that continue to select for enhanced survival. All of these are fighting against the chemotherapeutic regime that is being used to combat them. There's a number of strategies that are currently being used to overcome this type of drug resistance or cancer resistance. Combination therapies, where you're targeting more than one cancer mechanism by using a couple of different drugs and sometimes more. Using adaptive and personalized therapies, so you can try to treat a specific subpopulation based on a genomic profile. You're trying to exploit cancer vulnerabilities. Cancers do have weaknesses. If you can find out what those are, those can be targeted. And there's alternative delivery mechanisms as well to try to enhance drug efficiencies. These can include packaging the drug in nanoparticles, so the particles get taken up into the cancer cells, and the cancer agent stays there. Antibody drug conjugates are another widely used approach. So there are ways to combat cancer. There are many new approaches that are being evolved. And we very much hope that this has been a little bit of an educational process for you. It is quite incomplete, but hopefully it will have given you a little bit of an oversight onto the overall cancer process and its biology. Matthew?
Thank you. if folks, if you don't mind just unmuting yourself, let's give Paul just a little round of applause. Thank you so much, Paul.